Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar titled Achieving Scalable, High-Yield Plasmid DNA Purification Processes. I'm Alison Gash, an editor at BioInsights, and joining me today for the presentation is Klemen Božič, who will explore how to overcome key bottlenecks in plasmid DNA downstream processing. After the presentation, we'll have a live Q&A session. We invite our audience to pose their questions using the Ask a Question box at the bottom of your screen, and we'll try to get to it during the session. Now, I'd like to introduce our presenter. Klemen Božič is a scientist in the mRNA pDNA Process Development Department at Sartorius BIA Separations. Klemen holds a master's degree from the University of Ljubljana in Slovenia. He has extensive experience in pDNA analytics and currently focuses on continuous improvement of pDNA downstream processes. So, without further ado, I'll hand over to Klemen to kick off today's presentation.
Thank you for this kind introduction, and of course, welcome everyone to today's webinar titled Achieving Scalable, High-Yield Plasmid DNA Purification Processes. I'm Klemen Božič, and I'm a scientist in Plasmid and mRNA Process Development Team at Sartorius BIA Separations, Slovenia. Our facility is located in a small city, Ajdovščina, in the center of beautiful Vipava Valley, where we are surrounded by mountains, sunny vineyards, and many natural attractions like caves, water springs, etc. You are all invited to visit us. This webinar highlights the benefit of using Sartorius process solutions in downstream processing of plasmids. We will start with an introduction and then focus on presentation of plasmid downstream processing case study from cell lysis, clarification, chromatography, UFDF to final sterile filtration. Focus will be on test results of well-established and innovative Sartorius products used at different steps of plasmid production. Overall process performance with analytical and recovery results will be shown at the end. Plasmids are crucial in advanced therapies, particularly for vector construction and genetic information delivery. With the increase in clinical trials and the approval of new products, the demand for plasmids is expected to rise significantly, necessitating efficient, reliable, and cost-effective production processes. To start with, let's remember what are main three applications of plasmids. First one is use of plasmid as a direct therapeutic agent, naked or encapsulated. This is when plasmid is used in gene therapy or as a vaccination. Second application is transfection of cells, which then produce adeno-associated viruses or lentiviruses. 3 or 4 different plasmids are needed in this case. The third application of plasmids is to serve as a template in vitro transcription reaction. In this case, plasmid is used in linearized form. For the last application, when plasmid is used as mRNA template, the quality of plasmid before linearization is crucial. Here we present 2 articles which highlight the importance of plasmid purity and impact of potential impurities on quality of mRNA produced in IVT. Both articles declare that higher amount of impurities was observed after IVT if poorly purified plasmid was used as a template. For example, presence of open circular plasmid before linearization may impact the quality of the linearized plasmid and consequently the quality of mRNA produced using such template. This is a situation where our novel selective hydrophobic interaction chromatography approach for polishing of supercoiled plasmid can become an invaluable solution. You will hear more about it later. The study I will present was executed as a collaboration between process development team from Sartorius BIA Separations and Sartorius filtration specialists. It shows that Sartorius, besides producing outstanding chromatographic and filtration tools, can completely support customers at process development through cornerstone services. Our goal is to simplify plasmid production and purification process and to reduce cost of goods, so it is crucial to be ahead of market by anticipating market needs and executing extensive in-house research and development. Process development consists of three phases: process setup, process optimization, and scale-up. When it comes to process setup, we can rely on our extensive know-how, long-term experience deploying DOE approach, our cutting-edge technologies, and innovativeness. In process optimization phase, we deploy high-throughput solutions for screening. For example, 96 well plates offer a simple way to test different chromatographic parameters, such as loading conditions, elution conditions. Small-scale monoliths are used at this stage from 1 to 8 ml, and of course, the process is monitored by rapid at-line analytics, which is represented by our PATFIX Plasmid platform. Final stage of cornerstone process development service is scale-up and tech transfer. Plasmid production faces challenges like achieving high yields in fermentation process, processing of large volumes, establishing efficient chromatographic process for large and shear-sensitive plasmid molecules, preserving the integrity of supercoiled isoform, and the end, separating plasmid from impurities and achieving high recovery rates. There are many opportunities for optimization in plasmid production. First process to be optimized is upstream, as higher plasmid yield results in lower amount of biomass needed to produce certain amount of plasmid, less impurities introduced in purification process, and lower process volumes. But today we talk about downstream processing. Standard Sartorius BIA Separations plasmid production workflow is shown at this scheme. Alkaline lysis of resuspended cells is followed by calcium chloride treatment and clarification. Clarified lysate may then be prepared for DAE capture via UFDF or simply by dilution with water. Plasmid eluted from DAE column is then polished by hydrophobic interaction chromatography. As such, this is when pure supercoiled plasmid is needed, or after linearization, in case of plasmid template for mRNA, and if plasmid purity after DAE is of course sufficient. After concentration and buffer exchange, plasmid is sterile filtered to obtain final product. So how can we optimize the process? By lysis optimization. We can do this by replacing batch lysis with inline lysis and by applying DOE approach to improve lysis conditions. We can improve lysate clarification, separation of flux from liquid part that happens after alkalizator lysis increases filter capacity and thus reduces costs. The second way how to improve clarification is to do the filtration continuously, so being performed in parallel with lysis. This reduces hold-up time in harsh conditions and therefore the possibility of plasmid degradation. The next step to be optimized is tangential flow filtration, in case it is a part of your process. You can improve it by using appropriate TFF device and by optimizing process parameters on small-scale TFF devices. Regarding the plasmid capture, we strongly encourage our customers to avoid especially overnight storage of plasmid sample until capture step is completed. Continuous capture can solve this problem. After the capture step, plasmid is expected to be very stable. Plasmid polishing can be optimized, for example, by applying selective hydrophobic interaction chromatography instead of standard bind dilute. And at the end, sterile filtration can be improved by selecting the appropriate sterile filter to reduce product loss. Today, I will present the plasmid downstream processing case study performed last year at our site, utilizing Sartorius equipment and consumables, including several innovations. It was a pilot-scale study with a goal of producing 1 gram of pure supercoiled plasmid. This slide presents the broad list of equipment and consumables from Sartorius portfolio that were used at different stages of plasmid purification process, from automatic cell lysis system alkalizator to different types of filters, TFF membranes, and pilot-scale chromatographic system Resolute Multi-Use RCC that was used to perform chromatography on Simultus DAE and C4 HLD columns. The entire plasmid downstream process is monitored by Headline Analytics using PatFix pDNA platform, which is a powerful analytical tool comprising analytical columns, plasmid standards, customized system, and methods, all together in a user-friendly format. It is a rapid analytical chromatography solution capable of analyzing all types of samples, from lysate to purified plasmid, without need for difficult sample preparation. Only dilution of sample with binding mobile phase is sufficient. Benefits of Headline Analytics with PatFix pDNA platform are comprehensive insight into sample quality, meaning that separation of host cell RNA from different plasmid isoforms is achieved, enhanced faster process optimization, and monitoring the entire plasmid downstream process. It was roughly 4 years ago when we realized that to produce plasmid DNA on a gram scale, continuous lysis is the way to go. That's why a new machine, alkalizator, was developed by Sartorius BIA Separations. Here we list main milestones of alkalizator development. First continuous lysis experiments on prototype started in mid-2021. Initial tests were soon followed by kilogram runs in April 2022. Less than a year later, in beginning of 2023, first beta alkalizator was already installed at customer, and in December 2023, installation of first cGMP unit took place. On this slide, we can see also a picture of the alkalizator tower and the 5-liter lysis reactor. What are the challenges when scaling up alkaline lysis? It's batch-to-batch consistency, current lysis processes are not scalable, difficulties in controlling lysis time and managing large volumes, maximization of plasmid recovery, and, on the other side, minimization of plasmid damage. Our solution is automated inline continuous lysis, which ensures precise and monitored lysis control. Control systems allow adjustment of lysis conditions. It offers improved robustness and contaminant removal, is easily scalable, single-use, and CGMP compliant. At this picture, we can see how alkalizator improves separation of flux and liquid in comparison to the standard batch lysis. Here, the flux are all around the lysate material. On the pictures below, we see that liquid part of lysate is clearly separated from the flux, and we also see some differences in the flux composition when different airflow rates were applied to produce the lysate. And how the alkaline lysis using alkalizator actually looks like. Resuspended bacterial cells and lysis buffer are separately pumped into the first mixer, breaking down cell walls. The lysate continues through the coil, defining the length of lysis, is then neutralized and precipitated by calcium chloride in the second mixer. Air is added for turbulent mixing, reducing viscosity and capturing air into the flux, which makes them less dense and therefore they can float on the top of the liquid. Ultrasonic sensors ensure precise control and monitoring. After lysis is finished, the reactor is removed, components disposed of, and the system is ready for next operation. In our case study, we lysed approximately 700 grams of E. coli cell paste. We processed that material in approximately 100 minutes with a 5-minute lysis time and produced 35 liters of crude lysate containing 1.6 gram of plasmid. Importantly, this plasmid was very homogeneous, more than 96% of supercoiled and approximately 1.4% of genomic DNA contaminant. An example of analytical chromatograms for two samples of lysate is shown here. Red trace represents the sample after alkalizator lysis, green trace is batch lysis. Looking at the table, we can see that sample from alkalizator contained relatively more plasmid, less RNA, less genomic DNA, and less endotoxins than sample produced in batch lysis, confirming the efficiency of inline lysis. Very important fact is that sample preparation for chromatographic analysis consists only of centrifugation to remove cell debris and dilution of supernatant in binding mobile phase. Such diluted sample is then directly injected onto the CMAC pDNA analytical column, and after 9 minutes you already get an insight into plasmid quantity, composition, amount of host cell RNA, and plasmid. There are 1 liter and 5 liters lysis reactors available. Both can be operated using same alkalizator tower, which offers flexibility and scalability to meet diverse production requirements. Current maximum capacity is up to 50 kilograms of cell paste to be processed in 8-hour shift. Crude lysate has to be clarified, which presents a great challenge due to low filter capacities for crude lysates after batch lysis. Separation of flux and liquid in that case is relatively poor, leading to need for a large surface area of filters and consequently higher costs. Filtration train typically consists of two filters: depth filter Sartopure PP3 in 20 or 50 micrometer version, needed to retain flux, with typical capacity of 50 liters per square meter, and as a sterilizing grade filter Sartopore 2 XLG with 0.8 and 0.2 micrometer pores is used, with typical capacity between 120 and 160 liters per square meter. In case study, using alkalizator lysis, which improves separation of flux and liquid part of lysate, filter capacity was greatly improved. For depth filter, capacity was almost twice higher, whereas for sterile grade filter, results showed 60% higher capacity than usually observed. Besides filter capacity, also numbers for plasmid recovery are high. More than 93% of total plasmid was recovered. To get an overview, here is a schematic view of the case study from clarification step 1. More options for plasmid capture were tested. For the first option, we concentrated and diafiltered the lysate first. Two different types of TFF membranes were tested here. For the second option, lysate was inline diluted and loaded directly onto larger pilot-scale 800 ml DAE column. After DAE, L weights were pulled together and polished on C4 HLD column. To perform TFF for lysate, we used TFF devices with membrane area of 200 square centimeters. Optimal load volume was determined in a pre-study and was 4 milliliters per square centimeter. Here are some details about these trials. Despite slightly different behavior of both types during trials regarding flux and so on, both membrane types performed well. 300 kilodalton hydrosol allowed for faster processing. Nevertheless, it's important to know that 300 kilodalton cutoff may not be appropriate for smaller plasmid constructs, which would go at least partly into permeate. If we talked about general challenges in plasmid production before, let's now focus more on chromatography. Here we face lack of tailored solutions for large and shear-sensitive plasmid molecules, difficult separation of plasmid isoforms to produce homogeneous supercoiled plasmid, large load volumes and long operation times, and difficult removal of structurally related impurities such as RNA and genomic DNA. Zinc monoliths offer great advantages over other chromatographic media. High selectivity and resolution, enabling removal of unwanted plasmid isoforms, enhanced process productivity thanks to high recoveries, fast runtimes, and plug-and-play format, reusability and caustic stability, enabling cycling and reuse in development and in manufacturing. And last but not least, using monoliths means that process is easily scalable. Capture of plasmid was performed using simultaneous DAE monolithic columns. Besides plasmid capture, role of DAE columns in plasmid production is also removal of host cell RNA, reduction of proteins, and reduction of endotoxins. There are two types of DAE columns that can be used, depending on the process or user requirements. Each has its advantages. Simultus DAE with 2 micrometer channels offer high binding capacity for plasmid up to 7 grams per liter. They are suitable for plasmids below 8 kilobase pairs, are great for processing of lower volumes, for example when we have retentate after UFDF of clarified lysate. On the other hand, Simultus DAE with 6 micrometer channels offer low back pressure, allowing for high flow rates, and are therefore ideal for fast processing of large volumes, for example, for sample produced by dilution of lysate. They are suitable for all sizes of plasmid and for continuous process. Using both types of DAE columns for plasmid capture results in highly purified plasmid and high recovery rates. Since 2 different purification approaches were tested with and without TFF, we used also 2 types of DAE columns. After sample load, same chromatographic method was applied on both, consisting of flush with binding mobile phase to wash out the sample, wash with 0.6 molar sodium chloride to remove bound residual RNA, and dilution of plasmid using 0.9 molar sodium chloride. For 40 ml 2 micrometer column used for sample after TFF, plasmid recovery was 97%. In case of 800 ml 6 micrometer column, recovery was 95%. Importantly, more than 88% of eluted plasmid was in supercoiled isoform, despite suboptimal processing times at filtration step, where several different filter combinations were tested in approximately 3 hours. Analytics revealed good clearance of endotoxins, of proteins, and complete removal of host cell RNA. Purified plasmid eluted from the column in approximately 3 column volumes of 0.9 molar sodium chloride. CMAC pDNA analytical column can separate between host cell RNA impurities and different plasmid isoforms. Overlay of analytical chromatograms obtained by PatFix pDNA software reveals that both types of DAE columns 2 and 6 micrometer are equivalently efficient in removal of RNA, which is represented by blue and green traces, providing purified plasmid in the L-weight, which is represented by the red trace. In L-weight, we obtain 1.3 grams of pure plasmid with high integrity. As already mentioned, it was more than 88% supercoiled. Plasmid capture is followed by plasmid polishing by hydrophobic interaction chromatography. There are 2 aspects of polishing procedure that need to be considered. First one is quality of supercoiled plasmid. Polishing step has to increase the purity of plasmid by removal of impurities, like open circular plasmid, linear species like linear plasmid and genomic DNA, host cell RNA, endotoxins, and host cell proteins. They are listed here according to their increasing hydrophobicity. Supercoiled plasmid is slightly more hydrophobic than linear DNA species, and that's why it's quite challenging to separate them. The second aspect is process related. It's highly appreciated that process is generic to plasmid size, generic to amount of material, to load of impurities. On the other hand, it needs to be simple to scale up, environmentally acceptable, and GMP compatible. When talking about plasmid polishing, SIM C4HLD monolith is the best choice. There are several strategies or approaches of using this monolith for polishing of plasmids. Standard and still most frequently used is bind-dilute approach. Chromatogram of the left side shows how open circular and supercoiled isoforms of plasmid are separated in descending gradient of ammonium sulfate on C4HLD monolith. It's obvious that resolution is good even at 5 column volumes per minute flow rate. Besides high resolution, there are other benefits of monoliths, like high selectivity, high capacity, high flow rates allowing for fast processes, low shear stress, which is crucial for large biomolecules. They are multi-use and suitable for process intensification, for example, for cycling approach or simulating moving bed. Since bind-dilute is not capable of complete separation of open circular and linear DNA isoforms, including genomic DNA, from supercoiled, we developed a new chic approach utilizing lower concentrations of ammonium sulfate in load. Under these conditions, unwanted DNA isoforms go into flow-through during loading phase, while target molecule, supercoiled, binds to the monolith. This approach is generic to plasmid size, amount of and quality of load, and is easily scalable. It reduces the consumption of ammonium sulfate by 50% per gram of purified supercoiled plasmid. Importantly, using chic, product loss on pre-column filters is significantly reduced compared to bind-dilute approach due to lower concentration of ammonium sulfate. Chic approach was firstly extensively tested on small-scale monoliths on several different plasmid sizes, ranging from 4.7 to 23 kilobase pairs. Then we decided to move to slightly larger pilot scale, 40 and 80 ml columns. In our case study, we used 400 ml Simultus C4HLD column, which was connected to Resolute multi-use RCC system. 1.5 molar ammonium sulfate was used to bind the supercoiled plasmid onto the column. Since there was too much sample for one run, we simply divided it in 2 equal parts and performed 2 consecutive runs. Using chic, column capacity does not have to be exceeded to obtain desired results. This is a benefit of chic approach compared to sample displacement, where column capacity has to be exceeded to enrich the supercoiled isoform. More than 98% supercoiled plasmid eluted from the column, 1 gram in total. Recovery of supercoiled isoform was more than 90%, and endotoxins and proteins were reduced below the limit of quantification. Using user-friendly PatFix software, you can easily prepare different types of overlays, like grouping signals by signal type, by sample, etc., depending on what you want to investigate. Here we see an overlay that reveals the efficiency of chic approach on impurity removal. Black trace represents load, blue trace is the flow-through containing open circular and linear isoform, and red trace belongs to enriched supercoiled plasmid from L8. Plasmid sample after polishing step contains approximately 0.9 molar ammonium sulfate, which has to be removed by TFF. We tested 2 different chemistries of membranes for this purpose, hydrosart and polyether sulfone, both with 100 kilodalton cutoff. Someone may ask why not 300 kilodalton if it enhances faster processing? The answer is that in salty conditions, like after hydrophobic interaction chromatography, plasmids have more compact structure, requiring using such lower cutoff to prevent product loss in permeate, and this is also the case for large plasmids. Membrane area used was 200 square centimeters and load volume was set to 6 ml per square centimeter. From the result table, it is obvious that both membrane types performed similarly regarding the flux and processing time. Total plasmid recoveries were 99% for both types. We can conclude that both types of TFF membranes may be used for final UFDF of plasmids. The last step in plasmid production is final sterile filtration and filling. Sartorius has developed a new type of sterile filter called Sartopor Evo, which is a combination of the built-in 0.8 micrometer pre-filter in front of 0.2 micrometer final filter. Its innovative modification of polyether sulfone membrane further minimizes the adsorption of nucleic acids and proteins, making it even better in comparison to previous types of PES filters. This was also confirmed in our case study, where plasmid recovery after sterile filtration was practically 100%. Filter capacity should be more than 1,300 liters per square meter, calculated based on 90% flow reduction. Here we see also analytical chromatogram of the sterile filtered sample, showing that no additional open circular isoform was produced throughout ultrafiltration, diafiltration, and sterile filtration processes. We will move now to overview of study results. This table lists results for total and supercoiled plasmid recovery for each process step in our case study. Majority of steps show more than 90% supercoiled plasmid recovery, excluding lysis and first UFDF, where sample processing times were suboptimal due to additional trials that were performed. Overall process plasmid recovery was found to be 63%, which is an impressive number compared to some recovery data from competitors. Besides chromatographic analytics for sample quantity and quality, we performed also analytics for proteins by qubit, endotoxins by LAL test, and content of genomic DNA by dPCR. Analytical results speak for themselves. Supercoiled integrity is almost 100%, and all other impurities were found to be close to or below limit of quantification, therefore meeting all US FDA criteria. And now we are getting close to the end of the presentation. To conclude, here are take-home messages of today's talk, divided in 3 main goals of each plasmid downstream process, which are patient safety, cost per dose, and speed to market. When we talk about patient safety, we can say that alkalizator delivers lysate with high % of supercoiled plasmid and low genomic DNA contamination. It's important that simultaneous DAE columns efficiently capture plasmid and remove host cell RNA. Simultaneous C4HLD column enriches supercoiled plasmid and removes genomic DNA, endotoxins, and proteins. As a result, all FDA quality parameters for plasmids are met. Regarding cost per dose, end-to-end downstream processing of plasmid shows 64% of supercoiled plasmid recovery, which is a really high number. Alkalizator contributes to increased filter capacity by clear separation of liquid and flux in lysate, Sartorius XLG shows high capacity for clarified lysis filtration. Tumultuous columns and Sartorius slice membranes show high recoveries for plasmid. Tumultuous columns are multi-use and suitable for process intensification approaches like cycling SMB. Sartopor Evo as a final filter provides high capacity and yield for plasmid sterile filtration. Regarding speed to market, combining automated cell lysis with flow separation, clarification, and inline plasmid capture permits continuous processing, crucial for efficient large-scale production of plasmid. We can conclude that presented Sartorius process is a perfect choice for customers who are looking for scalable, high-yield plasmid DNA purification process. And we came to the point when I would like to thank everybody for joining today's webinar.
Amazing. Thank you, Klemen, for that fantastic presentation. We're going to start the Q&A portion of the webinar now. So if you have any questions that you want to send over for Klemen, please use the Ask a Question box at the bottom of your screen, and hopefully we'll get to it during today's session. So the first question that we have for you, Klemen, is what do you use for measuring pDNA concentration in the lysate and in-process samples?
Thank you for the question. So it was mentioned several times in the webinar, in the presentation, that we use our PATFIX Plasmid platform. So it's a chromatography-based analytics utilizing CMAC pDNA column, which is customized to get a high resolution between impurities that are present in the lysate, and at the same time also the resolution between plasmid isoforms. This is chromatographic analytics is let's say a basic or a basis to get an insight into the complex samples such as lysate, and so you need to separate all the impurities if you want to quantify the material. We do perform also. We do measure also the quantity of plasmid. We can do that by plotting the area from the chromatogram to a standard curve for plasmid, which is prepared using in-house plasmid standards that are also part of the, let's say, analytical platform. So you can buy standards of different sizes, use them as a material for a standard curve. There are two of them, one for, let's say, around 5 kilobase pairs, and the second one is 15 kilobase pairs, and then you can, depending on the size of your construct, plot the areas to any of these standard curves. For maybe in process samples, so when they're more pure, then we can also use the nanodrops or UV-Vis spectrophometry. But for the initial samples, that's not it's not accurate. Practically, you cannot get an insight in the quantity of nucleic of the plasmid in the lysate using UV spectrophometry.
Great. Thank you for that answer. That was very in detail. We have another question here. Is it possible to adjust the duration of lysis and ratio between different solutions used on alkylizatol?
Yeah, it's very adjustable. So as I mentioned, there are 4 pumps, 4 peristaltic pumps that pump the different liquids, different buffers into the lysis reactor and other mixers, so different parts of reactors, and all these flows can be adjusted to the one you want. So you can change the time of lysis by changing the flow rate. You have, of course, let's say 1 liter reactor, and by reducing the flow rate, you will increase the contact time of your cells with the lysis buffer, and vice versa. And also, of course, you can use different types. We have, let's say, customers that want to use their own lysis solutions. Maybe it's just different concentration of some buffer, or it's completely different chemistry. It can all be used and adjusted according to the customer's need.
Thank you. And we have another question. What are the quality requirements for pDNA when used as a raw material and not as an API?
It's a good question. It's actually, at least to our experience, up to customer. I understand this question. So in a way that if you use the plasmid as a template for mRNA, or as part of, let's say, 3 plasmids to produce viral vectors. So I would say that at least same standards or same requirements should be applied as when plasmid is used as a API, to be on a safe side, let's say, because we can say that it's pretty, the criteria are pretty loose. If we take a look into the criteria for super cold integrity from the FDA, it's up above 80%, which is really not, let's say, very strict criteria. As I mentioned at the beginning of the webinar, if you have, for example, 15% of open circulars, which is nicked plasmid, you can not say exactly what will happen with this plasmid after linearization and after that plasmid is used in the IVT, even if it's completely okay according to the FDA criteria. Similarly, for the plasmids used for the transfection, so a higher percentage of super cold is, let's say, connected or has been recognized as a reason for better transfection efficiency. So being closer to 100% is always better than being on the lower limit. And same for the other, let's say, impurity-related criteria. So genomic DNA is definitely not something that you want to, let's say, have in your material, even if not being used as an API. And also for the other impurities, proteins, and the toxins. So if you remove them completely, then you are on a safe side, and you can do it by, for example, using our purification process.
Well, thanks. Yes, a fantastic answer again. We have another question from our audience. How do you determine percentage SC and OC pDNA in your samples?
Yes, thanks for the question. Again, I'll return to our analytical workhorse, if we call it. It's also the chromatography analytics with PATFIX or plasmid pDNA column. It's capable of separating plasmid isoforms. There are 2 types of column. the same as for the for the preparative purpose. Also for the analytical purpose, there are let's say a column which is suitable for smaller plasmid, and the one which is which should be used for larger plasmid. The 1.4 micrometer channels provide better resolution between plasmid isoforms, so you can separate open circular from linear, and then the next isoform that elutes is supercoiled, and after supercoiled you get also concatenars, so dimers, trimers, and so on. 6 micrometer channels, which are used for larger plasmid, do not offer such a high resolution, but still you have a resolution between open circular, linear, and supercoiled, but multimers are somehow, let's say, hidden in the tail of the supercoiled. But definitely you can. Yeah, the answer is by chromatographic analytics. We do determine the percentage of isoforms.
Fantastic. Yes. We have another question as well from the audience again. How much material can be processed in 8 hours using alkalizator?
Yeah, so the max capacity of the alkalizator in 8 hours, calculated by applying 2.5 minute lysis time, is 50 kilos, and that should be processed on a 5-liter lysis reactor. Yeah, so up to 50 kilos at the moment per 8-hour shift. So that is at the moment, but yeah, depending on the different recipes or something that customers want to apply, that may change.
Fantastic. And yes, another question. What timescales are you describing going from lysis to polishing?
If this refers to the case study that I presented, so we were working on a pilot scale. We can say that we started the lysis or the resuspension of cells, let's say at 9 o'clock in the morning, and the capture step was finished, including all these trials that were performed in between. The capture step was completed roughly at 3 in the afternoon, so 6 hours to get a material in the after the TAE capture step. And still, yeah, there are, I would say, at least an hour and a half of that could be spared if processed really in optimal conditions. So yeah, this is what, as I mentioned, we encourage customers to do, to process the material up to capture step, because after capture step we performed so many times the storage of DAE eluate in the refrigerator, not even in the freezer, in the refrigerator, and not changing its composition, the composition of isoforms at all. So maybe 1% or 2% of open circular may be formed in half a year, but yeah, but up to that step, the degradation rates are incredibly high, and it's really worth putting the efforts into fast processing and therefore maintaining high integrity of your target product. And then, yeah, the process for the polishing can be done immediately after the capture. You simply adjust the concentration of ammonium sulfate, and you can start load it. If you have the system and column ready, 2 runs were performed in an hour and a half, I would say. So really quick processes.
Fantastic. Thank you again for that detailed answer. Our next question, what are the recovery rates for pDNA in each process step, specifically for both chromo steps?
Yes, maybe I went slightly too fast through the table, but the recovery rates are summarized in the table. And if we talk about the chromatography, we had 2 capture steps, and we had recoveries around 95 or 97% for 2 different types of DA column, and then Syn4HLD resulted in 90% of recovery for the target supercoiled plasmid, 91%. So yeah, we are always above 90% of recovery. And also, yeah, for the other processes, as I mentioned, the first, let's say, if we talk about supercoiled, we had slightly lower recoveries of supercoiled because of degradation in first process steps, but otherwise this recovery would be higher also in that steps.
Right, fantastic, thank you. And another question from our audience, why would you advise not to hold the plasmid overnight prior to capture stages? Is it a question of yield or product quality?
Yeah, maybe I partly answered that question before, but yeah, again, we also heard about cases, even if let's say it's hard to believe that the plasmid remains stable also overnight with almost no degradation in such harsh conditions. But according to our experience, we do see significant degradation. We performed a lot of studies. Yeah, fortunately, I did not put the example chromatogram here. So we stored the lysis at let's say an aliquot at room temperature, an aliquot in a refrigerator at 4 degrees, and another one in a freezer. And it was in all cases we observed the degradation, which was of course the highest at room temperature, but either in the refrigerator the percent of open circular increased at least for 50%. I mean, in comparison to the starting percent of open circular. And what does that mean? That means that you lose your target product, which is very precious, it's expensive, and why not get as much as possible from your starting material if it's only related to how you will run your process. So if you can set a process that allows you for high recoveries, why not doing that?
Fantastic, thank you again. Very, very detailed answer. We have time for a couple more questions. So is SHIC approach applicable to different types and sizes of plasmids?
Yes, so selective hydrophobic interaction chromatography. I think I mentioned in what it was tested on broad spectra of different sizes, from I would say 3 kb up to 23 kilobase pairs. So it's really generic to plasmid size. These were plasmids of different types, so from mRNA coding plasmids to helper plasmids, AAV plasmids, just some model plasmids used for the standard. So yeah, in all cases it performed well. So yeah, this is really generic. I can say that we did not had a case where this approach would not be applicable. So Yeah. So
Amazing.
okay.
Yep, that sounds fantastic. And we have time for one final question. Can we reuse the columns for different pDNA? Have you identified any residual pDNA after processing various plasmids?
Yeah, in principle, each construct should be purified on a separate column to prevent any cross-contamination. So the cleaning procedure should be validated in case you want to use the same column for different constructs. This is not something that we can provide. So you can reuse the column several times for the same construct. That's not a problem. So that's why we declare that columns are multi-use. And but using the same column for different constructs, I would say it's up to the customer to stand behind such approach.
Fantastic. Thank you, Klemen, for answering all of those questions. Unfortunately, that's all we've got time for today, but any questions we didn't get to, we will reply by email. This webinar will also be available on demand tomorrow, so look out for an email from us with the link. We'd also like to invite you to take a short survey to provide us with feedback on today's webinar, the link to which can be found in the resources on the right-hand side of your screen or in the on-demand email as well. Additionally, an attendance certificate will be available on the right-hand side of your screen. All that's left to do is to thank Klemen once more and to thank you to the audience for watching, and we'll hope you join us again soon.
Thank you.